Okay, so welcome to all of you. Today, in our journal club meeting, we have Dr. Murthy, and Dr. Murthy is a doctor turned neuroscientist who is currently serving as an assistant research scientist at the Laboratory of Cognition and Emotion at University of Maryland College. And he's going to talk about the slow and fast gamma oscillations in human EEG beyond laboratory, and which are going to be his insights from Alzheimer's disease research. So I would request all of you to welcome Dr. Murthy. And yeah, I think we are good to start. Dr. Murthy, if you could take over.
All right, thank you all. I guess you could all hear me.
Yes, we can.
Yeah, thank you. And thanks for being here on Sunday, like 6:30 in the evening. Thanks for attending my talk here. So I'll start sharing my screen. So can you people see my screen? All right. So yeah, thank you for a wonderful introduction, by the way. So uh, I'm currently working from home as a postdoctoral associate at University of Maryland in the laboratory of Professor Luis Pessoa, and um, I'm also involved with a small startup with a friend who is also present in the talk today. Thanks for coming. And this whole talk, I'll be talking about most of my work which I have done during my PhD days at the Neuroscillations Lab headed by Professor Supratim Ray at the Center for Neuroscience at Indian Institute of Science, Bangalore. Okay. So, to be honest, like this is the first talk that I'm giving online to a large bunch of people whom I can't really see. So, if you have any questions, just feel free to interrupt me and ask. And if you're like feeling sleepy, also just tell me. I'll try to change my pace. If you're not understanding anything, just let me know. Okay, just and if you have any doubts, also you can mark them up. You can ask later on, or you can also stop me and ask while we are having the presentation. Okay, so and this presentation, I'll be talking about slow and fast gamma oscillations in human EEG. That will be the most, that will be my focus for the talk today, as how I have gone on to discover this something really nice phenomenon in the brain called as slow gamma oscillations. Because this whole world of gamma itself is quite weird and interesting at the same time. And when it gets slow, it gets even more interesting. And I'll also talk about some insights that I have gained through Alzheimer's disease research, as well as research in healthy aging population, which I'll present towards the last part of my talk. Okay, so so let's begin. So as I have said, we are going to discuss electroencephalography today. Pretty sure you have come across this word in previous talks, especially when in the first talk when sleep was being presented. So to be um, so we all know that there are various neurons in the brain which help us in perceiving and understanding the world and responding to it, right? And these neurons talk to each other by means of ionic or electric currents. And we can actually measure these potentials by placing electrodes at various levels inside the skull or inside the cranium or outside the skull, right? So we'll see both levels of recordings today, where you have electrodes where the skull has been removed in animals, and electrodes have been advanced inside to record the field potentials, which are called as local field potentials, pretty much because these are local. And we'll also see, for the most part of the talk, we'll be seeing these potentials being recorded from outside the scalp. We place electrodes outside the scalp like this, as you can see in the figure, and and you record the electrical activity that is coming out of the brain. So this is quite non-invasive procedure called as electroencephalography, where you don't have to really do anything to the subject except putting some small amount of conductive gel, which is biocompatible. And this is pretty much analogous to your ECG, which is done on your heart. The only difference is that in EEG you have like very large number of electrodes. In my case, I have used around 64 electrodes for recording. Okay. For a historical perspective, EEG was first described by this German psychiatrist called as Hans Berger. He observed that the moment you place electrodes on the scalp of a person and ask him to close his eyes, you see some waves like these. These are pretty much oscillatory phenomena, which you are seeing in the potentials being recorded in EEG. And when the subject opens his eyes, these oscillations go away and are replaced by faster activity. So Berger wanted to name these, and instead of saying these as Berger rhythms, he said alpha. And when the subject opens his eyes, he called that activity as beta activity. And and alpha is rightfully known as Berger rhythms, in fact. Now, I'll give you some examples of oscillations in EEG that I have recorded. This is an EEG signal that I have recorded in our lab. It's pretty much a clean recording, and it looks like this, different channels, and you can see voltages across time for each channel. And if you take these time domain waveforms and run them through certain signal processing techniques called as Fourier transforms, for example, you can represent the power at each of these electrodes not in terms of time, as you see on the left side, but in terms of frequencies, as you see on the right side. So let me, yeah. So for example, uh, this is your time domain waveform. You run a transform on this called as Fourier transform, and here it was amplitude, or just you can also uh, uh, substitute with power if you square it, for example. So on the y-axis here there is power, but on x-axis here there is time, and here there is frequency. That's the difference between these two types of representations. It's the same signal which is represented in different domains. Here it is the time domain, and here it is the frequency domain. And as you can see here, the power in the lower frequencies is high, and it is steadily falling exponentially as frequencies increase. But at the same time, you also see a nice bump here, which is around 6 hertz. If you place a crosshair here and measure what frequency this is, it will be around 6 hertz. So this falls between 4 to 7 hertz roughly, and we call this as theta oscillations. Similarly, in another signal, you have the same 1 by f, the exponential decay of power with frequencies, and you also have this nice little bump around 8 to 12 hertz, peaking at around 10 hertz. And this is your alpha rhythm. And this is exactly what Hans Berger has described in his initial papers. And in the plot that I'm showing you here, it yeah, this works. So this is actually, you can see this oscillatory phenomenon happening here. This is actually your alpha rhythm. All right. Over time, various rhythms have been described in EEG, like for example, delta, theta, then you have alpha, beta, etc. And in this talk, as the title mentions, we will be concentrating mostly upon gamma rhythms. And the way I'll be taking this is like presenting a short story about gamma, like how it evolved, the beginnings, and how it has evolved. And then I'll talk to you about the weird thing called slow gamma, the new thing called slow gamma. And then we'll see how these gammas can be tapped in aging populations as well as in Alzheimer's, and what they have to offer us, what they have got to offer, right? So, so when we talk about gamma, who discovered gamma? There are three good papers which mention about gamma almost at the same time. So one is at 1987, then 88, and then 89. It's not that one discovered the gamma earlier to the other or whatever. I mean, they have done this research independently and almost simultaneously. It's just that during the process of publication, sometimes things get delayed. And so the credit of discovering gamma actually goes to Freeman, but Econ and Gray have contributed equally in describing gamma rhythms. And in this talk, I'll be presenting some results from this paper, which will help you understand what exactly is gamma and how they have done this. And please remember that all these research have been done in various animals, either monkeys or cats. Okay, and so these are not EEG recordings. These are recordings using depth electrodes. So they have advanced electrodes into the cortex by removing the scalp temporarily, and then they have recorded these signals. So this is one example. So when we talk about these depth electrodes, these so the suppose your animal is viewing the entire visual space. Okay, somewhere in the visual space the animal is viewing, and your electrode will be active if something happens at a particular location at that space. Okay, that particular location is called as receptive field, and it usually is spoken about about the electrode. It's a property of the electrode. So, for example, Charlie Gray was recording from one particular electrode, and when he presented a particular stimulus on the receptive field of that particular electrode, that electrode was very happy and it started giving out various signals crazily. Okay, so for example, you look at this stimulus. This is a bar. This area is your receptive field of that electrode, and whenever a bar was moved in a particular direction, this is a time period when the bar was moved. Okay, in this time period there was no bar, and here that bar there was a bar, and then again there was no bar. Okay, so he started recording certain spiking activity from the neurons which were around that receptive field, around that electrode in the brain. Okay, so these spikes are nothing but the activity of neurons in particular when they talk to each other. And then when you also record very low frequency signals from the raw data that is collected, you see that there are voltage fluctuations that get recorded. Okay, these voltage fluctuations are nothing but your local field potentials. Okay, so when this bar was present, when this bar was absent initially, you had certain signature in the local field potential, which was slower in rhythm. But when the bar was presented, this all of a sudden this potential started getting noisier, right? Getting more active, let's say. Similarly, even in the spiking activity, you see that there are a lot of spikes when the bar was presented compared to when it was absent. Okay, so this is a bar of light, by the way. So Charlie Gray is showing this figure with a zoomed in version also. So when you zoom in this little portion in both the traces, the upper trace looks like this, which is very nice oscillatory structure, and the lower trace looks like this, which is spiking activity. Okay, and if you look at this you know the scale bar, you'll notice that this is around 20. So you'll have one cycle for around 20 milliseconds or so. So converting that into hertz, you'll get a frequency of roughly around 40 to 60 hertz or so. Okay, so this is called as gamma activity. It's a simple thing. You are activating a certain region in the visual cortex, which it really likes. That particular region in the visual cortex likes this bar a lot. And when you present that bar, you start seeing gamma activity in the local field potentials. Okay, so this is again a amplitude versus frequency plot, which is also called as a power spectral density plot, if it were power on the y-axis. And here you see that in the control condition, when there was no bar present, this was flat like this, around 20 to 60 hertz. But when the bar was presented, you see this bump. So this is increase in power or amplitude in this case, increase in amplitude from 20 to 60 hertz range. And this is nothing but your gamma power. Okay. And so David Hubel and Torsten Wiesel have done like really brilliant experiments, which have won them Nobel Prize. And in these experiments, Hubel and Wiesel say that the spiking activity is actually dependent upon the orientation of the bar that you're presenting. So if it is like this versus horizontal or vertical, etc., the spiking activity will change. The rate of spikes will change. Now, Charlie Gray says that this is true even with gamma oscillations that you measure from the local field potentials. So, for example, if 0 is degrees is the orientation that your electrode likes a lot, and if you label it as 100% amplitude, then as you move away, as the orientation moves away from the preferred orientation, the amplitude starts decreasing on either sides. Okay, so this is orientation dependent. So it is dependent on the stimulus. It has to be a stimulus that the visual cortex, that region likes. So in this case, it's a bar, and it has to have a proper orientation so that the gamma recorded is maximal. So they suggest that gamma oscillations are actually stimulus dependent. Okay, they say that gamma oscillations are stimulus dependent. So now, after this brief overview, let's see what happens. Let's see the important questions that we'll be discussing in this particular talk. So I don't want to bombard you with a lot of information, and we'll try to build a story that leads us to slow gamma oscillations. And so I have cherry picked a few papers which are like really seminal in this field, and I'll be presenting a few figures from these papers for you to understand. Now, if you feel any difficulty, just stop me and ask questions, right? So there are these questions that we'll be looking into, like how is this gamma recorded from LFP different from the spiking activity, and whether this gamma has an upper bound and a lower bound. Or so we say that 30 to 80 hertz is gamma. Okay, so whether it's just 80 hertz or whether it can be more, or whether it can be lesser than 30 hertz, this is a question that we'll be asking in the next 10-15 minutes. Okay, so this is a seminal paper, a very important paper, which talks about the differences between gamma and spiking activity. This is coming from Alexander Thiele's lab, and Gieselman is the first author. So Thiele is at University of Newcastle in the UK. Okay, so it's a fantastic lab, by the way. And here, what they have done, this is in 2008. So the earlier papers that I have shown was in 1998, and over the 10 years, there was a lot of research, 10, 20 years. There was a lot of research regarding the mechanisms of gamma, etc., etc. And this one has changed the way, what we think about gamma in a lot of ways. So here it's a small stimulus, and these stimuli are grating stimuli. If you can see, these are alternating dark and white bars, similar to what Charlie Gray had presented, but these bars are numerous in number. Okay, so these kind of stimuli are called as gratings. And these gratings were also, again, this is also a monkey experiment. And when these gratings were presented to the monkey, and specific electrodes were recorded from the visual cortex, what Gieselman observed was that when the grating was small, then you had these spikes. So this is called as a raster plot. Okay, and this is very similar to what you see here. Okay, these are all your spikes. So at every spike, with every spike, you put a tick. Okay, and that is this tick. And the y-axis actually represent the number of trials that you have that tick. Okay, that you are recording from. So this vertical bar represents the stimulus on. So your the small stimulus was present between this duration, the duration between the two vertical bars. Okay, so the moment these vertical bars were presented, you see that the spiking activity, which was otherwise very less before the bars were presented, started getting ramped up. Right, you're seeing a lot of spiking activity in these regions, and that's what also represented in this histogram, which denotes the spiking rate. That it was very less in the beginning, and once the stimulus was presented, you started seeing this spiking activity. And again, when the stimulus was absent, when the stimulus was removed, the spiking activity persisted for a few milliseconds, and then later on it decreased. Okay, so but you don't see any oscillatory activity in the local field potentials that you're recording from the electrode. Whereas when the size of the grating increased, then this spiking activity reduced in rate. The spiking rate got decreased, but if you can see here, the gamma oscillations have become more and more prominent. Okay, now this is also seen in this curve. How do we say that this is gamma? By simply taking Fourier transform, and you see that for the larger grating, which is lighter gray, the gamma power was high, and as you go to the black grating, the gamma power was almost negligible. So with increase in size of stimulus, gamma power decreased. Oops, okay, this got exchanged. So with increase in size of stimulus, gamma power increased in power and spiking rate decreased. Okay. So that's the takeaway point from this paper. Now coming to the next study, this is another important study which has brilliantly separated the high gamma activity from gamma oscillations, and it said that high gamma activity is something very similar to spiking activity, and this was carried out by Supratim at John Mansell's lab at Harvard Medical School. So Mansell is at University of Chicago now, and Supratim is at IISc. So what Supratim did was he took similar gratings, and he saw how these gratings would change the gamma activity as well as the activity which is above like 100 hertz or so. Okay, which is called as high gamma activity in literature. And what he noticed was that for small gratings, high gamma was quite prominent, as you can see in this figure. But as grating diameter increased, high gamma activity reduced in power, strength, and gamma activity started showing up. Okay, this kind of a plot is called as a time frequency plot. So till here you have seen just amplitude versus or power versus frequency. Here you have frequency and time, both information. So frequency is on the y-axis, time is on the x-axis, and the color represents the change in power from a baseline period. Okay, so 0 is again same as in this plot. 0 is the onset of the stimulus, which is your grating, and the stimulus was present till 400 milliseconds. Okay, so you can see that as long as the stimulus was present, both the high gamma and your gamma activity were present. Okay, so they were absent in the absence of the stimulus period, in the absence of the stimulus. So they were primarily driven by the stimulus. Now, the difference between the two is that as the grating size increased, high gamma activity reduced in power, and gamma activity, which is this nice band of activity that you're seeing, so this is the 50 hertz mark, right? So this is from like 40 to 60 hertz or so. It's a very narrow band of gamma, very beautiful activity. So this activity increased in power this confirms the earlier study by Giselman and Thiele. The only difference is that this portion, the high gamma activity, is very high for very small gratings, whereas for the same set of gratings, this blue curve, gamma activity is negligible. And gamma is very high for this orange curve, which represents the largest diameter grating, but its high gamma activity is very low. And this is kind of quite correlated with your firing rate of the spikes. Okay, so Supratim concluded with also with so these are like just correlational analysis. I mean, very simple analysis. You just say that as spiking increases, high gamma also increases, or as spiking decreases, high gamma decreases. So both are correlated with each other. Now, Supratim has in fact done quite complicated analysis that I won't show here, which suggested that high gamma and gamma are two different phenomena with different mechanisms of generation and mechanisms of or with different origin. And so indirectly, he has put an upper bound on the gamma activity per se, which is around 80 or 100 hertz. Gamma can't go beyond that. Okay, it's so whatever is beyond that is high gamma activity, which is correlated to the spiking activity and not like real oscillations. That's what that paper indicated. Okay, now so till now we have been looking about studies in animals. Now we'll briefly look about studies in humans, and then quickly go to what slow gamma is, right? So this is a study in MEG. This is one of the earliest studies. You can see the year here. It is 2006. So this is almost after 18 years or 19 years that gamma was explained in animals, that in humans, that too in magnetoencephalography, gamma was explained in humans. Okay, so so you can understand how difficult it is to see gamma activity in humans, and this is because you don't have the luxury to open the skull and directly record from the brain. And when you record from the skull, you'll get all sorts of noises, especially the environmental noise or the noise of the equipment and things like that. So it's not easy. So yeah, I mean, you have to be very careful when someone says gamma is fantastic and it's there in humans, and lots of papers claim, but you have to be careful when you try to understand gamma. Okay, so this is MEG. MEG is nothing but the magnetic counterpart of EEG. It's although it's like 20 times costlier than EEG. It's actually 3 times costlier than MRA. So if so, it costs around I don't know 20 crore rupees or so. So in India, you have MEG only at one place, and that's at NIMHANS. Okay. And this is one of the earliest papers which spoke about such narrowband gamma oscillations in the visual cortex in humans in MEG. Why do I say narrowband? It's because if you see in this plot, this is like really broadband. It extends all the way from like 90 hertz to 240 hertz or 300 hertz or so. So it's very broadband. But this gamma is narrowband. It extends from like 70 to maybe 80 hertz or so, 65 to 80 hertz or so. That's it. It's narrowband. Okay. And in EEG, also, what people did was they presented these grating stimuli, small grating stimuli to humans. And this is one of the earliest findings. So it's in 2013 again. Just look at the years. Okay, so one of the earliest findings in EEG, you see this gamma band at after zero, I mean the start of the stimulus. Okay, and it was localized to the posterior electrodes, which are more active to your visual stimulus. So all of you know that your visual cortex is towards the back of your head, the occipital lobe, right? So that's why you see the activity of gamma when the stimulus is presented at the back of your head. Okay. Now, so to summarize this portion, we saw that gamma was discovered around 1987 to 1989, and then it was said that these rhythms are tuned to the orientation of the bar or the grating that you have presented. Okay, and then in 2008, it was said that while gamma increased with the increase in size, spiking activity decreased. And this spiking activity was actually correlated with high gamma, more than 100 hertz or so, putting an upper bound to the gamma that you can see. But you still don't know what the lower bound is. People always would think, based on these bumps, people would think that the lower bound is around 30 hertz. 30 to 80 is what they have been saying as gamma all throughout. But let's see. Okay, we still don't know what the lower bound is. And in humans, so these were done in animals, and parallelly in humans also recordings were done using EEG and MEG, and they have seen the gamma activity. Now the question is, what happens if you change the size of the stimulus more than what you have seen in the earlier papers. So what happens if the stimulus size is increased further? For example, instead of taking this little grating here, what happens if you cover the entire monitor screen with the grating? Okay, that's like really interesting. The reason why I thought of that is that I thought, okay, it's very difficult to record gamma in EEG in humans. So why not just inflate the stimulus so it will give you robust gamma? That was a bad thought. I don't know how bad it is, but then that's like serendipity. It works. And so what I did was I took a lot of subjects and showed them these full screen stimuli. So the entire screen is covered with these various different kinds of gratings, different kinds of bars, okay, or with different orientations and different contrasts, different sizes, etc. So what I found was that when the size was largest, that is the full screen grating, I still saw this nice gamma band. Okay, this is between like 40 to 70 hertz. That's what I saw. And I saw that increase in the power spectral density plot also. The black curve represents the baseline condition when there was no stimulus present on the screen. And the pink curve or the magenta curve represents the condition when the stimulus was present on the screen. So you definitely see an increase in power between 40 to 70 hertz. Okay, and you see that activity again in the occipital cortex. So sorry, yeah. The interesting thing or the surprising thing is this. So when I showed this full screen stimuli, I started seeing this nice bump, which was between 20 to 40 hertz, as you can see in this figure, in this plot. Okay, the magenta trace indicates when the stimulus was presented. Okay, this trace is from the signal when the stimulus was present on the screen, and you see that it causes not only this the traditionally known gamma rhythm, but also a newer gamma rhythm, which I started calling a slow gamma because it was slower in frequency than the other rhythm, which I started calling fast gamma. So now we have two gamma rhythms in human EEG, not just one. Okay. The first one I've called as slow gamma and the second one as fast gamma. All right. And both these gamma rhythms were most expectedly seen in the occipital regions, which is not surprising. So, okay, now that we have one more gamma, we were like extremely happy. The idea, my basic thesis was about Alzheimer's disease, and it was very difficult for us to record gamma oscillations in humans, especially when we looked at previous papers. So now, when we were thinking like what to do, suddenly something happened, and we got to know that there is just not just one gamma, but there are two gammas with which we can work. And we were like really excited about that. And what I actually noticed in the lab was that the size of the gratings increased, the power in both the fast and the slow gamma increases. So this upper plot is for an example subject whom I have shown here also. The lower plot is average across subjects, and I had like 15 subjects. Which is not a very bad number. Um, but I'll tell you next, like what exactly is a bad sample size. All right. So this is average across like 15 subjects. And this one is just one subject, example subject for you to see. So if you see here, the important thing is that for the 8-degree grating, you see slow gamma here, but you don't see any gamma here for the 4-degree grating. However, you see fast gamma for the 4-degree grating. So with 4-degree gratings, which people have used in the previous experiments, all experiments of humans and animals, just concentrating on a particular location of the visual space, they could actually see fast gamma. And all that they had to do was to increase the size of the grating, the diameter of the grating to 8 degrees. They had to double this diameter, that's it, to find another gamma, which they have missed for almost 25 years. Okay. And for me it was a bingo. And as you can see from this plot, as you increase the size of the stimulus, the power in both the gamma oscillations increase. So the basic concept remains same, that these oscillations, both the oscillations, are dependent upon various stimulus properties, such as the orientation of the grating in monkeys. I'll show that next. So size, for example. And I also wanted to do this experiment on monkeys. So I suggested Supratim, let us do this on monkeys. Okay, and at that time I was like extremely busy getting data from human subjects. Throughout my PhD, I had collected data from around 300 subjects by myself. And later on, another person has helped me collect another 100 subjects. So we actually in our lab during my 4-year period from 2016 to actually 2015 to 2019, both of us have ended up collecting around 400 subjects data, EEG data. Okay. Um, Now this one was monkey data, and as you see, you see that the fast gamma was tuned to orientation, as you have seen in previous studies. Like the first GRACE study, you've seen that gamma oscillations in LFP were orientation tuned. It was true for slow gamma, for fast gamma that we have seen. Similarly, for slow gamma also it was true. So slow gamma in for this monkey was maximum for grating of orientation of 0 degrees, and it almost vanished when the orientation was 90 degrees. And we have recorded from 2 monkeys almost around like more than 100 electrodes, both the monkeys combined. Okay, and you see that the black trace is for slow gamma, the gray trace is for fast gamma, and these have different tuning curves to orientation, which is one of the first proofs which we give that these two are different oscillations, in fact. Okay, so slow gamma is different from fast gamma. That's the this is the first proof. The next proof comes from this, question that whether slow and fast gamma are generated by different networks in the brain. And what we saw here, this is the 8-degree grating case, that slow gamma was, the power in slow gamma was very negligible in the initial few milliseconds of the stimulus presentation, whereas fast gamma was quite prominent in the first few hundred milliseconds, and the power seems to be decreasing as time passed. And this is exactly what we found when we quantified this. You see the green curve, slow gamma is slowly climbing up in power for the 8-degree grating case, and fast gamma is slowly declining in power for the 8-degree grating case. So we postulated that these two gamma oscillations are generated by different networks in the brain. We still don't know what these networks are, but it's a speculation that it can happen. Okay, and these networks have slower time constants for slow gamma and faster time constant for fast gamma. That's what we postulated. And the happiness is when your work is verified by other labs, okay, and especially in other animal models. So we have done this work in both humans, which is my work, and in monkeys, which is the work of my friends Vinay and Pooja. So Vinay is at UMCP, he's a postdoc there in Shihab Sharma's lab. And Pooja is a PhD student now at University of Chicago. Okay, so all three of us have contributed equally to this work, by the way. And uh, I'll skip this for now. So the happiness is when your work gets certified by other labs, and this is that it's one such example where they had used mice viewing such gratings, again alternating dark and white bars. Okay, and the grating size increased, and as the grating size increased from 8 degrees to 60 degrees, you start seeing that fast gamma decreased in power and slow gamma increased in power. Okay, so black is the 60 grating curve and gray is the 8-degree stimulus. Okay, so you see slow gamma also in mice. This is work by Julia Waite from Hillel Adesnik's lab, and we had actually submitted these papers almost at the same time. So, and we had spoken to each other during our, when we met at SFN conference, Society for Neuroscience conference at San Diego. So we had spoken about our results. So this is like really interesting, and they have gone one step further and also delineated the mechanisms of these gamma oscillations. Okay, so they say using various optogenetic techniques that I won't get into here, they say that then there are two distinct networks, one formed by the parvalbumin and pyramidal cell network. These are, don't worry about these names, okay? These are just neurons in your brain. Pyramidal cells are excitatory neurons, and parvalbumin and somatostatin neurons are inhibitory neurons, okay? And they say that fast gamma is driven by this network, which is quite local. So when you have all gratings present, it will be driven very well. And you have lateral connections with the SOM neurons and pyramidal cells, which form another network. As the size of the gratings increase and SOM neurons start getting covered by the gratings, covered into the stimulation, then SOM also, the inhibition from SOM kicks in, and it starts showing the slow gamma oscillations. So this is a beautiful synthesis which they have supported using optogenetics, and yeah, that's where this is. So we have, so to conclude, when a grating stimulus is shown to a human subject or anyone, animals also, you have this black trace as the baseline power spectrum, and there is no stimulus present on the screen. But when the stimulus is present, you have one bump, which is your slow gamma oscillation, and then the other bump, which is fast gamma. And these oscillations are driven by different networks, and that has been proved at least in mice. We don't have the luxury to prove the same in humans or monkeys, because you can't. There are only certain things that you can do with humans and monkeys, right? So you can't do optogenetics, at least as of now. It's not possible. Although people have started to experiment in monkeys. So that brings us to the end of the most major and the most exciting work, which is it's exciting because it is new. People have never thought of this before, and this has opened up an entirely new arena of work, which is your slow gamma. Okay. Now, briefly, I'll take you through. I mean, obviously, if you have slow gamma and fast gamma, what are you going to do with it? Okay, we have it. Then what? The answer is, when you come out of the laboratory, you go into the community and see how these oscillations are different and what help can they give you in the clinics. Okay, so in the past, people have suggested that the gamma, which is now fast gamma, okay, because it is between 40 to like 70 hertz. This gamma decreases in frequency, becomes slower as age advances. That's what people have suggested. Okay, and they have done this experiment from all the way from 80 years old children to 45 years old adults. So what I asked was, what happens after the age of 45, or maybe after the age of 50? And this is the result. That from 50 to 64, so this is in the young adults, the same age group as here. And you see fast and slow gamma in males and females. Upper row is male, lower row is female. As age advances, 50 to 64 and more than 64 years, you see that fast gamma decreases very well. Okay, and this effect is quite prominent. Slow gamma also decreases. You see that, but you don't make that out in these TF plots. So you see that here. Okay, so what I have done is I have taken all the power values for subjects aging from 50 to 88 years old. Okay, and there are total of 227 subjects in these plots. And you see that the change in gamma power for both the fast and the slow gamma is negatively correlated with age, and it is significant for both slow and fast gamma. So even if you correct this using Bonferroni, this will be significant, right? And you see the same in the scalp plots. So these are your different electrodes with different power values, and the redder it is, more is the power. And you see that these power values are highest in the occipital electrodes, as you have seen in the previous plots. And as age advances, these become less and less redder. These become more and more yellowish. So it's just saying the same story as in these plots. Okay, and it's not just the power of gamma which decreases with age, also the center frequency of the gamma power decreases with age. It is negatively correlated, and it is significant for both slow and fast gamma. So you can see that in these plots as well, change in power plots, that these bumps start moving leftwards as the color goes from darker gray to lighter gray. So darker gray is your this age group, 20 to 49. Lighter gray is more than 64 years. So as age advances, you start seeing that the curve moves leftwards. Okay. And okay, we'll skip about alpha power. Don't worry about alpha, because we'll concentrate only on gamma for now. So what happens when you have, so this was all about healthy aging, all right? You have to, this is very important. So even if the even if the volunteers were 88 years old, they were healthy, cognitively healthy. Okay. And even then you see that gamma decreases with age. Now, in MCI and AD, so MCI is mild cognitive impairment. This is thankfully this group has more of doctors compared to engineers. Well, I'm not thankful, just joking, but for those who don't know these terms, AD is Alzheimer's disease. Okay, it's a disease of the elderly in which there are various dementing symptoms and signs. Dementing, mental means your mental processes get weaker. Dementing is your mental processes in general get weaker. One of the earliest symptoms or signs is your memory weakness. Okay, so Alzheimer's, you shouldn't confuse dementia with memory weakness. These two are different things, and dementia is an umbrella term which includes more cognitive deficits. The earliest is your memory deficit. And mild cognitive impairment is a preclinical stage of dementia, which has a propensity to progress to Alzheimer's, or it can stay as MCI also. It can stay like that throughout the subject's life. So there could be cases where MCI can get better also. It's not. So MCI is a preclinical stage which does not come to your hospital, and that's one reason that we don't see if we want to look at MCI people, you have to go to the community. You can't get those samples in the hospitals. Okay, and that's the reason that I had to record from me and my friend had to record from 350 subjects. And given, fortunately or unfortunately, I don't know, the prevalence of MCI and Alzheimer's in India is very low. So MCI is about like 4 to 5%, and Alzheimer's is 2.5% or so. So out of the 300 subjects, what I got was only 14 subjects who are MCI and 6 subjects who were AD. These are all taken from the community. We went to, the recruiters went door to door, every apartment, and they said, oh, we are doing this project, please come and join us, be a part of it. It's a very huge project, which involved 15 different PIs in 3 different centers in Bangalore. Okay, and this is how we got subjects. And sadly, because this was a community project and not a hospital-based project, we had just 14 plus 6, 14 MCI and 6 ADs. Total was 20 subjects. And we were able to manage. So I'll show you the results now. If you see, the fast gamma and slow gamma both seem to be reduced in the MCI AD group compared to the healthy individuals group. Okay, and this is like really striking because in Alzheimer's in the early stages, so this is 14 out of 20 is MCI, preclinical Alzheimer's, right? So in the early stages of Alzheimer's, you don't see any symptomatic deficits in vision. All that you see is with memory impairment, as I have said. But functionally, there's something wrong going on in the networks. We don't know what it is. It's a speculation. We have spoken about 2 different networks, SOM and parvalbumin networks. So we don't know what is happening. But in the preclinical stages, it is possible that these networks are getting deranged, and somehow the brain is trying to cope up with such functional changes. That it is being able to maintain the function of visual cortex at least till the late stages of AD. However, you can see such changes through functional mapping using EEG. That's the whole point that is really exciting to me about this whole slide. Okay, so you see that change quite well. I mean, this is a Kruskal-Wallis test. Given that the sample size is so less, I've done Kruskal-Wallis test, and you see that for the MCI-AD group, the power, the slow gamma as well as the fast gamma power is very less compared to healthy subjects, and it's quite significant KW test, right? And you see similar same things with the scalp plots also. Now, Unfortunately, when I separated NCI and AD, I saw the same trends, but these trends were not like really significant. The reason was the small sample size. Okay, which you can see again here. It's just because you have so less samples. But if I have to increase my sample, so I've submitted this to a prominent journal, and that's the main question that they have raised, whether I can do more samples. Unfortunately, I can't, because one is because of the COVID situation, and the other is if I have to do another 300 subjects to pull out 6 ADs out of them, it will take another 4 years for me, right? To summarize, with healthy aging, power and frequency of both slow and fast gamma reduce with age in healthy elderly. And this is important that it happens even in healthy elderly. Okay. And in MCI, also this happens compared to their age in gender matched healthy subjects. So if it is happening in healthy elderly, if the gamma power decreases in healthy elderly, it decreases even further for the MCADI group, provided you take this limitation into account, the small sample size. Okay. To summarize, to stitch all of this together, changes in gamma rhythms during aging may represent a continuum of healthy aging NCI-AD spectrum. It's a continuum. So it was stable in the initial, during the adulthood, and then during healthy aging, it started declining. And if for example, if the subject has preclinical or clinical disease, Alzheimer's, then the decline is even more. So it's a continuum that you have to keep in mind. And why this is important, it is because there are lots of fraudulent research which is going on in this particular domain, where they're trying to come up with therapeutic benefits of artificially entraining your neural oscillations at the gamma rhythms, so that the subjects can become better. Okay, this is one work which is done in Lee Huisite's lab. They call this as genus gamma entrainment using sensory stimuli. So what they do is I'll take just 2 more minutes. It's almost done. Huh. So they have these, flickering kind of patterns. There's nothing here. It's not a grating. It's a flickering light. And they have kept these mice in these flickering lights or flickering sound also, for example, as you see in this particular figure, either flickering light or flickering sound at 40 hertz. What you see is that that flicker is captured by your brain signals. So you start seeing 40 hertz rhythms in your brain, which is about your gamma rhythm. So gamma is 30 to 80. 40 is just one number within this range. It's a very peaky rhythm. It's not a narrow band that I have shown you. It's a very peaky rhythm. Okay, it's quite different from what we have discussed so far. But they say that entrainment at that frequency, which falls in the gamma range, causes betterment or reduction of amyloid load or the reduction of Alzheimer's pathology in the mice models. And this is Professor Lee Huisai at MIT who is demonstrating her idea, which is very happy about it. So when you're doing these kinds of experiments, you must be careful that you're taking the age factor into account. I'm pretty sure that these are pups with genetically modified pups. So they start showing Alzheimer's at early stages of their life. But when you're trying this out with humans, you have to be careful. Okay. And and another last thing that I have found was that these entrainments, which we call as SSVP, these entrainments decrease with healthy aging. Okay, this is like extremely important. And unfortunately, because of the small sample size, we didn't find any difference in the brain entrainment at 32 hertz between the normal subjects and the healthy controls group. sorry, the healthy controls and the NCID group. So whatever Professor Liu Sai is demonstrating here, or in this genus paradigm, this is seen even in normal healthy aging, and unfortunately we can't make a very valid claim that it does not it is comparable in NCID and healthy controls because of the small sample size. But if we have more sample size and the result holds true, then whatever Professor Lewis has demonstrated will have to be reworked, and we'll have to understand what's the difference between the mice work that they have found and the human work that we have been doing. Okay, so to summarize, you have this human work. So in mice and humans, you had this slow gamma and fast gamma described about the same time. Here I have left out another important paper in neuroimage. It's an MEG paper which was published around the same time, and it has also replicated slow and fast gamma oscillation. The first author is Pentazis. Okay, I'm very happy that there are 3 labs, 3 different models, animal models, which have come up with the same oscillatory phenomenon, which is slow gamma. And then Li Huisai and her students, Singer, Maturel, and others, Ayakarino, others, they have suggested that entraining mice with gamma oscillations reduces the Alzheimer's pathology in brain. But what I have seen is that such an entrainment, it gets decreased even in healthy aging. So it's not specific only to NCI or AD population. It gets decreased even in healthy aging. And so are the slow and fast gamma oscillations. And actually, in Alzheimer's research, our group is the only group which has done this in humans. The visual narrowband gamma oscillations. We have discussed the results before. We don't know what the future is going to look like, because Liu Huisai's team has applied for a patent which got even approved last month for clinical trials in humans, clinical trials of this genus paradigm. It will be very interesting to test whether they'll be successful or not. I'm actually hoping them to succeed, because it will answer the pains of a lot of elderly population around the world. And plus, what I suggest is we have to test this particular paradigm where you have static grating stimuli shown to mice. Okay, just like what Wait et al. Have done. Mice which are Alzheimer's models, and see if gamma has any therapeutic benefit there. So I have not, nobody has really asked me any questions till now, and I hope that I'm still connected to the internet.
Yes.
Yeah.
You do have a lot of questions in the chat box. We'll bring it up after you.
Really? Okay. So yeah, I want to acknowledge and thank all my lab members. Supratim was a wonderful mentor. I mean, any amount of words for him will be very less, actually. It's quite a wonderful mentor. And Vinay and Pooja have been great friends who have helped me with the monkey experiments. And Kirtana is fantastic. She has left the lab only last month, and she has joined University of Pittsburgh online for her PhD. And then there are others who have helped me in a lot of ways. And this is the Tata longitudinal study for aging. This is a multi-center project which I was talking about. These are all the people with whom I have worked to get this thing really done. I'm really thankful and grateful for all of them. So yeah, I think this is the questions time. I don't know. I haven't checked. I didn't have the opportunity to check my inbox.
Yeah. Thank you, Dr. Murty. So I had a question also.
Yeah.
So in the case of the gamma oscillations, and you were doing Fourier transform to get those gamma oscillations, there are one type of waves which are present when a person is in the awake state also, right? The beta waves, if I'm not wrong.
Okay.
So so even if you're doing Fourier transform, wouldn't the theta waves interfere with your calculation of the gamma waves?
No, so when we do Fourier transform, it is done frequency wise.
Yeah, yeah.
So your theta or beta or alpha, all these come before gamma. So there
Okay,
is no way that they'll interfere.
okay.
I mean, see, if you're talking about harmonics, etc., then that's a different question. So if you look at this particular slide, okay, you see that this is your alpha rhythm, right?
Yes,
And when the stimulus is presented, this alpha rhythm actually goes down. So
yes.
there is less chance that you'll find a harmonic of this alpha at around twice its frequency, which happens to be the slow gamma.
Okay,
Okay.
okay. So we have a
Uh,
question from Sabha, and I would request Sabha if she's still in the meet to come up and ask, and if she's comfortable. Otherwise, I'll ask. Yeah. Okay. So her question, anyways, is what do these gamma rhythms, the
uh,
gratings, tell us about Alzheimer's? What conclusion can we draw from these graphs about Alzheimer's?
okay. So as I have said, you so this particular graph shows that the gamma power, both slow and fast gamma, decreases with preclinical Alzheimer's and Alzheimer's put together. So in this plot, I have 14 preclinical and six Alzheimer's patients combined together. Okay, so they make about 20 subjects, and in the next plots, these are separate for MCA and AD. So so when we show these gratings to Alzheimer's or MCI people, it is more probable that you'll notice lesser gamma rhythms compared to their healthy age and gender matched controls. Okay, so so it's a probability that I'm talking about. As of now, we have not done any clinic, so that's why I said that these are like insights. These are my insights, right? So we have not done any rigorous testing in the clinics or any sites where we go to the hospitals and take Alzheimer's disease patients and run this. So if I have to really answer your question, we have to perform these experiments across different continents, just like how your vaccine trials happen. Okay, these are like diagnostic tests. Okay, which we haven't done as yet. But what insights that I get is that is reduced in these states of preclinical and clinical dementia. Okay. Now, whether the interesting question here is whether it is seen only in Alzheimer's, whether it is specific to Alzheimer's. It does seem to be sensitive in Alzheimer's because it can catch the preclinical condition. But is it specific to Alzheimer's? That is not something that we know of. We have not. So this low gamma stuff has come only when we published our papers, right? In 2017, Wait et al. 2018 was Fantasis and me. So this whole business has started to come up only in the recent 3-4 years. So we don't know whether these effects that we are seeing here are specific to Alzheimer's. Okay, there more people will do these tests, more people will read our results, and more people will do these tests in different neuropsychiatric conditions. I hope that answers your question. Hello.
Yeah.
Yeah.
Uh,
Okay.
okay. So the next question is by Vivek Sharma. Uh,
Uh,
Vivek, would you like to ask?
Okay. Hi. Thanks for the great
hello.
talk. I was
Yeah.
just
Thanks a lot. Yeah. Hmm.
uh, the kind of trend that you show with the gamma frequency is somewhat similar that we saw in the alpha frequency as well. So I was wondering, did
Uh-huh.
you like that? Is there any kind of trend that we see with the alpha gamma coupling?
Alpha gamma coupling is not something that we have tested. So see, so what is the trend in alpha that you have seen?
Like the shift in the
Frequency, right?
the age and the shift in the frequency. Yeah.
Shift in the frequency with age. So the problem with our analysis is that we have taken 500 millisecond chunks of data. So that for the Fourier transform that has given us only 2 hertz of revolution. Okay. So your alpha, for alpha, we have only 3 points, 8, 10, and 12 hertz. So with these 3 data points, we can't really comment about how the alpha frequency shifts. Okay, and hence we also cannot comment about any sort of coupling per se.
Right. Right.
So
Okay.
it's a
Thanks.
limitation in our study. Yeah.
Yeah. Okay. Thank you.
Okay, so the next question is by Shri Harshini, and if you could come up and ask Shri Harshini.
Hi, sir.
Hi, hello.
My question was, did you find any differences in the power of the high gamma and the slow gamma between males and females of the same age group?
Yeah. So I guess by high gamma you mean fast gamma.
Oh, sorry, yeah,
Okay.
fast gamma.
Yeah. So that's a fantastic question. Actually, I had I didn't put that up, but what do you think could be the answer? I mean,
No,
I'm curious to know like why you have
yeah,
asked
when
this
I
question.
see the graphs, it
Ha,
was
ha, ha.
power was more in the females.
Yeah, exactly. So this is again something that I have seen that for fast gamma, if you look at this, especially this is true for especially in the younger age group as well as more than 64 years age. The fast gamma as well as slow gamma seem to be higher in females compared to males. Okay, and yeah, we haven't spoken about these, so I won't confuse you with these now. But in, importantly, these results have also been seen in another EEG study, EEG or MEG, I don't remember that, by Whittingstall's group in Canada. Okay, this was a neuroimage paper which they published in 2019. We had me and and Russell Butler, who was the first author, had a brief discussion about this, and they believe that it is because of the varying skull thickness in males and females. But I'm not sure. That has to be really tested out rigorously. The reason is that all these power values are computed from baseline, right? So you
Yes.
have some baseline power, and against that you compute these power values. So if these power values are affected because of skull thickness, then your baseline will also be affected because of skull thickness. So I don't know how skull thickness is important. And another thing is that if skull thickness dominates these kinds of patterns, then in older, as you age, your skull becomes thinner. So
Yes,
as you
sir.
age, you should start seeing more and more gamma power, which is not what happens, right? You start seeing less and less gamma power. So we don't know. We actually don't know. But it's a very good question that males and females have different levels of power, and that's quite significant. And wherever I have presented this till now, at UCSF and at conferences, people have started saying, "Hey, females have something really special going on." So I try to avoid this, gender-based discussion during this talk. But I'm glad that you have brought this up.
Thank you, sir.
Thanks for a wonderful question. Yeah.
Okay, next question we have by Disha. Disha, if you could come up and ask.
Yes, good evening, sir. First of all,
Hello.
thanks a lot for this amazing talk.
Thank
So,
you.
I think my question is quite previously answered, but I would put it right now as well. So, do you think can this method be used for diagnostic purposes? So, as you said that it can also detect in the pre-Alzheimer's condition as well. So,
Huh.
can is there a possibility of using this in diagnostic purposes of
Definitely, it
Alzheimer's?
is. It is. There is a possibility. That's what That's my insight that I have gained from my PhD work. But the only thing is, if I were to use this as a diagnostic test, I won't in the clinic. Why? Because this is just like the Russians coming up with their wonderful vaccine. Whether you're going to take it or not at this stage is a question, right? It's the same question here. So this has not passed through various clinical tests that it should have had undergone through, right? And we don't know what the validity of the test would look like, what would be the accuracy, the sensitivity, specificity. So my research is a step one in this direction, and there are like lots of steps to go ahead. Uh, uh, but I strongly believe that it can definitely classify normals from some abnormal people. I don't know. Should be able to classify.
Yeah, thanks a lot.
Yeah, thanks.
Okay, so the next question is by Aditya.
Sir, good evening, sir. First of
Hello.
all, it was a very informative meeting.
Thank you.
Okay, my question was that we know that beta waves or beta oscillations occur during attention, and alpha when we open our eyes, and alpha waves occur when we close our eyes but are still attentive.
Mhm.
So, what is the probable reason for gamma oscillations for a grating stimulus? And can you also give some other examples for stimulus that cause the similar gamma oscillations?
Oh, that's a fantastic question that, um, I thought I'll put it up, but I thought I'll keep the pace slow. And I also thought that I don't want to bombard you with a lot of information. But you have to, so which background are you from? Are you a medical student, or do you have some background in math? Do you understand
Uh,
basis sets?
yes, sir. I'm actually a second year med student here in
Huh.
Madurai Medical College.
Oh, that's so nice. So, do you understand basis sets? Okay, let me tell you this way that all your visual scene in visual scene you have like different components, right?
Yes, sir.
Uh, so all these components can be decomposed into smaller and very smaller components.
Okay.
And these components, when you look at them, these are actually these gratings.
Okay.
Okay, you add, subtract, multiply these gratings in different possible combinations, and you get these beautiful visual scenes that you see. Of course, I mean, when you use achromatic gratings, black and white gratings, you'll obviously get black and white scenes. There's no
Okay.
doubt about it. So there have been instances where people have seen that gamma is produced even for natural images. Okay, like for example, a tomato or a black and white car. Okay, so what people have suggested is that when you are recording from a specific neuron inside the brain of monkeys, which are seeing these images, when grating like structures fall on the receptive field on these electrodes, then only you see gamma. You understand?
Yes.
So, for example, if you have a penguin which has a black hand, a white tummy, and a black back. Okay, it looks like black, white, black, alternating dark and white bars, right? Gratings. So when that particular structure falls on the neuron, on the retina, which responds to that neuron, then only that neuron or that electrode shows gamma oscillations, not otherwise. And these gamma oscillations are very specific. You see only if you shine bars or gratings and nothing else. And these gratings also have specific features. Okay, just like they're tuned to a particular orientation, they're tuned to particular spatial frequencies. So in my first, the third slide, yeah. So here you see these gratings. So these gratings are dark and white bars are crowded together. Here they are less crowded. So this is more spatial frequency. This is less spatial frequency, right?
Yes.
So you will see gamma only for this grating, not for this grating. So there is a certain limit of spatial frequencies with which you see grating, gamma, not otherwise. It's very specific, and it's annoyingly specific. And gamma is also produced for screens like these, which are completely red or completely green. No need of grating, but to color also they are seen, they are observed. we don't know, like, so Vinay has a paper in PNAS on this particular topic, and that effect has been seen both in monkeys and humans by different labs. So Vinay was the first to do it, and then there was another lab in Texas which replicated the findings. So natural images do produce gratings, do produce gamma, but it's not to the natural image that gamma is generated. It's that particular small grating-like structure in the image to which gamma is generated. Otherwise, you don't see gamma for natural images. It's very specific.
Okay.
Did I answer your question?
Yes, sir. Yes, sir.
Yeah.
And also, are they do the gamma oscillations occur in other stimulus such as auditory or sensation?
Beautiful question again. We don't know. So my junior, my friend, very good friend of mine in the lab is doing on auditory work. Maybe if you are interested, you can do something on somatosensory or some other area. We don't know. We've just unraveled mysteries about vision, both achromatic gratings as well as colors. Now it's the turn of auditory. So if you're interested, you can talk to that girl.
Yeah, sure. Thank you, sir. Thank you very much.
Yeah, thanks for the questions.
Yeah, on a similar accord, Pradeep and Shri Vidya are asking. So Shri Vidya is asking, why is the visual stimulus this particular grating? Is it because the contrast provides better readings? And Pradeep is asking, are gamma oscillations significantly seen only for visual stimuli, or they can be seen with other kinds of stimuli, which you partially answered right now?
Yeah. So I have answered Pradeep's question. I mean, we don't know. The reality is we don't know, and it's an open arena. It's a very interesting field of research now. Okay. So Srividya, do you? So I didn't get your question. So what do you mean by this particular grade contrast grating?
So my question was, I think you partially answered it already.
Uh-huh.
Ki, why? So we are trying to give a visual stimulus and induce gamma rhythms. So why is it that this peculiar kind of visual stimulus is generating these rhythms? Is it because of the contrast provided by the black and white bands? Is that what the neurons are responding
Yeah,
to? Or
that's true. So at the level of your retina, these, the cells, I mean, not the rods and cones, the ganglion cells, these respond well to contrast, not to luminance. Okay, contrast is diff of luminance. It's the difference between luminance at 2 points. Okay, so you have these on and off center grading. So when you should read about David Hubel and Austin Wiesel's work on this. And if possible, you can also go and read Hartline's work. Hartline was the first to suggest such experiments in retina, for which he was awarded the Nobel Prize. And later on, David Hubel and Austin Wiesel followed his path. So, and they unraveled these mysteries in the primary visual cortex as well as LGN. Okay, so the important thing is that in LGN, you have these on and off center cells, and you have these respond to such edges, not edges, but the contrasts. Okay, and when it comes to your visual cortex, these are no more small dots where you have differences in contrast, or small circles where you have different contrast. These now start, these neurons now start looking at bars, which are circles placed side by side. Okay, this is like feed forward connections, or the information transfer that happens from the LGN to the V1. Okay, that's how your percept is being constructed in the most early stages in your striate cortex. Okay, so these respond to bars, and by definition, gratings are like alternating dark and white bars, right? So that's the reason that your striate cortex goes extremely crazy when it sees these particular gratings and not otherwise. Okay, for that, if you want to understand this in more depth, you have to read Hubel and Wiesel's work. I mean, they have these 4 seminal papers. from 1960s onwards. Beautiful work.
Can you post the names of all these authors and papers on the chat?
On the chat,
Yeah, or
how do I do that?
You could just click on the
The chat. Okay. David Hubel and Austin Wiesel. Okay, so the good thing is that they have like good videos on YouTube also, how they have done these experiments and so you people can go through the videos also, and you'll hear those spikes. You'll actually experience those spikes. Yeah.
So Basudha is asking, does this also happen in other neurodegenerative diseases like Parkinson's, or is this
We
specific
have
to
no
Alzheimer's?
clue. As I've said, I mean, we have done experiment only in Alzheimer's till now. So we can't talk about specificity at all. And I don't see a reason why it should not happen with Parkinson's if it happens with Alzheimer's.
Okay. Uh, Alison is asking, do you think the reduction in gamma oscillations with age is related to pyramidal neuron loss, especially in MCI and AD?
Pyramidal neuron loss. So yeah, we don't know about this. So yeah, so there are some rodent experiments in which they have seen gamma oscillations go down due to abnormal interneurons, the parvalbumin interneurons. Like if a sodium channel is missing, and this is work of um, okay, I'll tell you what it is. So they have even tried to transplant properly working these interneurons into the brains of Alzheimer's mouse to rescue the function, for example. So it's more, they say that it's more related to interneuronal deficits. Now the problem with such studies is that they haven't really tested with gratings. They have always taken the baseline gamma power, and they have tried to make conclusions based on that. Okay, so that's a limitation of their study, but they can definitely try to do this with gratings and by inducing gamma oscillations, visual inducing gamma oscillations. Okay, so it's not, I don't know whether it's because of pyramidal cell or loss or not, but they suggest that it's because of interneuronal deficits. Okay. Does that answer the question? Hello?
Yes.
Yeah. Okay.
Yeah, she says thank you.
Okay.
Okay. So the next question is by Sai Dharshini. He asks, what is the functional role of slow gamma oscillation?
Ah, so yeah, okay. Thanks for bringing this up. And I'm pretty sure you might have had read somewhere that gamma is functionally significant. The thing is, it's a very big controversy which is happening around with the functional as well as structural relevance of gamma. Okay, and that's the reason that I haven't put it up. So if you look at my slides, there is a big gap between 1989 and 2008, and in it's not little, it's actually big. This one. So this is 1989 and then 2008. During this time, there have been a lot of studies which have explored the functional significance of gamma oscillations, and they say that these are correlated with working memory and binding problem. Binding is when you combine different pieces of information in the visual scene to create a visual percept. Okay, so they say that gamma is involved in all those mechanisms, all those functions, higher cognitive functions. But the truth is that there is no supportive evidence as on date which can attach a causal relationship between gamma oscillations and these functions. Sure, we have numerous papers which talk about correlations, Okay, but I have not come across any paper which talks about causation. And it's very difficult to test causation in humans or even in animals, because if you want to disrupt a function of the brain by, uh, if you want to disrupt gamma oscillations in the brain and then test whether the higher cognitive function still exists or not, you have to also disrupt the normal functioning tissue of the brain, right? So it's very difficult to test for causation. And unless there is any causation, it's wrong to claim that gamma has a function or alpha has a function. All these are just correlational, and we have no clue whether they have any function or not. And one reason I'm happy about it is that I don't have to look into functions. Okay, I can just see, I'm a doctor turned neuroscientist. All that I'm interested in is the diagnostic value of whatever I study, whether it will be useful to the public or not. So whether they have function or not, fine. I don't mind. But whether they're useful for diagnosis, I guess they are. That's all I'm happy about. Have I answered your question? It's a controversy, and yeah, it's a very big controversy. I mean, people fight, and people fight all the time.
Okay, so this is probably going to be uh, one of the last questions. So this
Sure.
Amandeep asks, does the power or the frequency vary with visuals accordingly? Is there a correlation? If yes, is there a difference in healthy versus AD sample?
Power, sorry, can you repeat?
Does the power or the frequency vary
Uh,
with visuals accordingly? Is there a
Visuals, what is
correlation?
visuals? Visual stimulus, is it?
Yeah.
Okay.
Is there a correlation?
Okay, okay. Yeah, I got it.
If yes, is there a difference in healthy versus AD sample?
Yeah, so as I've said before also, there are a specific set of spatial frequencies to which the gamma can be seen, spatial frequency of the grating. Okay. Similarly, as the contrast of the grating increases, the power of grating increases. And people have seen really interesting phenomena, like as size increases, the frequency decreases. As contrast increases, the frequency increases. All such phenomena. I haven't shown these papers to you, but there are like really interesting and seminal papers, both in humans and monkeys, to this effect. And the second part of the question that you have asked, whether these different gratings, whether that difference is also seen in MC and AD. Thanks for that question, because the reviewers have also asked me that question, and I'm working on it. So soon we'll have the answer for that. For now, I don't know, but I don't think so. To guess, if I were to guess based on my experience, I don't think that the difference between the stimuli will also show up in Alzheimer's. But it's a good question that I need to test. Does that answer your question?
Okay, so the next question is by Ankush, and he's on screen. Right.
Okay, thank you for the great talk.
Thank you.
So I have a few questions. One is, so when you showed us the gratings, there was a prominent blue line in the bottom of the graph. What do you think that was?
Yeah, that's a good question again. So it's good that I have skipped a few parts so you can ask questions. So have you seen this? I've spoken to this, right?
Yeah, yeah.
So yeah, you have the slow gamma, fast gamma, and here I've said that there is decrease in alpha power. Right? This is your alpha bump, alpha power in the baseline condition. And this magenta line is when the stimulus is presented. And as soon as the stimulus comes up on the screen, there is decrease in this power. Okay? It's called as alpha suppression.
Yeah.
Now, the same alpha suppression, if you were to see in time frequency plots, it looks like that. This is alpha suppression.
Okay.
Okay. So
this is not a alpha signal, right? This is
some
alpha
other signal. This is alpha signal, but it has gone down. So if you were to close your eyes, your alpha will be something like that. It will be even peaking. You were to close your eyes. You were opening your eyes with a gray screen in front of you. It's like this. And when something comes upon the stimulus, it reduces further.
Okay.
That's it.
So you mentioned center frequency in between somewhere. I could not understand what the center frequency means.
Yeah, center frequency is nothing but the frequency at which the peak occurs. So, for
Okay,
example,
cool.
this is your center frequency for slow gamma, this is for fast gamma, this is for alpha.
Makes sense. Okay, I have two more questions.
Sure, go ahead.
So, why do you think the fast gamma decreases as one goes older? Is there a certain explanation for it?
Why decreases? So initially I thought that it's because of structural changes that happen in old age, but there have been papers where they say that it's just correlational, it's not causational. So again, there is a bit of controversy here whether structure of the brain really affects gamma power or not.
Okay, a question
So
related to that.
No, if I say that, okay, it's not structure, it's some sort of neurochemical transmitter or something like that, that also there is no proof. It's just a speculation. There are studies which have looked into these aspects, but there is no causative relationship that has been given. So we don't know sure, like why it happens. We only know what happens.
Okay, one more question is, so do you think that can be because of training of neural networks with experience? I mean, so as we train our neural networks more and more, we have to use less of our brain areas to do a certain job, right?
Is it?
I mean,
is it going to be always the truth? Always the way? I don't know. I mean, it might happen that we're training, you're recruiting more and more neurons.
Yeah.
I mean, I don't know. I'm just guessing.
Okay. Thank you.
It's a good. No, I think it's a good question. I mean, if you're interested, you can take data from. You can join the lab and you can work on this. Yeah.
Thanks.
Yeah.
Okay, I think we're going to wrap up the questions now. The last question we are going to take is by Noshra, who wants to ask the question himself.
Okay.
Noshra, you can take it and then we'll wrap this up.
Yeah, thanks for your great talk. It was very informative. And for me, especially because I have a similar, I mean, I when I'm doing my project, I've seen similar results in the computational study that I'm working, which is the shifting of the gamma peak toward the lower frequencies. And the reason I found this in my studies is actually increasing the decay time of inhibitor neurons in a balanced network. And I just wonder if there's any experimental studies that support this idea, or anything that you want to add, or maybe
see. Actually, it's a good question, and so yeah, as far as I know, there are very less. So you can go through the work of Adam Kahn. Okay, Xiaoxuan Jia and Adam Kahn. So Jia was his student. Adam Kahn is at Albert Einstein College of Medicine, and they have done some very good work on this. So they have published papers in Neuron and Journal of Neuroscience in 2011 and 13. I don't know whether you have gone through those. The other thing is, there was one student in our own lab at Supratan's lab, who was working on a similar stuff, so computation as well as experiment. And what he found was that the center frequency of these rhythms would depend on the prior state of the network. So if the network is already in an excited state, then then the frequency would be different compared to what it was when it was not in an excited state. Okay, so you excite the network showing a grating with a very strong contrast, and then you show grating with 50% contrast. Then it's not just the power, but the frequency also falls down, something like that. So he has done this computationally, and he has done this in one monkey in our lab. And by the time the recordings were done, and he, the second monkey recordings were to start, he got a PhD offer from Adam Kahn himself, and he went there. And I don't know whether he's carrying out that research now or not, but he's a you can talk to him. It was a very good result that he has seen. So yeah, I don't know where that result has taken him to now. It isn't yet published. He's been there for 2 years now.
Mm-hmm.
Yeah. You can talk to him. So,
maybe.
Yeah. So it is Adam Kahn, and his student is Abhimanyu Liri, and they're at AECOM at NY. Okay.
Yeah. Thanks for your information. Thanks again for your very informative talk.
Thank you.
Yes, I think we can wrap up now. Thank you, Dr. Murthy, for your amazing talk, and thank you for being patient.
You can call me Murthy, you don't have to call me Adam.
Thank you for being patient throughout the questions and answering them.
It was my pleasure.
It was really informative, and thank you so much. Thank you so much to everyone who joined for today's Journal Club. It has been a wonderful event, and we hope that you will continue to be associated with us. Thank you so much.
Thank you.